With that in mind, we start with a pilot study where we select 5 individuals, 9 samples of those because we do sequential studies. And we pick on purpose as a proof of principle to test very complex cases like the one I just showed. Mostly the ones that we already have bulk sequencing and we know that they are a combination of mutations or they show parallel evolution. Other thing that was interesting to test was to go to our freezers and go to frozen PBMCs that have been there some over a decade and see if those were reliable to be used for this technology. The way that we used the Tapestri, and we picked this 34 gene panel that they have available, which actually include most of the putative driver genes that we see in CLL. So we took for this one. So this is the data, and we did this in collaboration with Mission Bio sequence the samples. So were used already the new sequencers. The number of cells that were used were between 5,000 and almost 13,000 cells. One in coverage varies, especially depending on the sequencer, but one very interesting thing that we saw was the uniformity. I think that we were very pleased to see that overall, and my colleague showed in detail, but that was another panel, but that overall has a very good uniformity, so in most of the amplicons are covered in enough coverage. This looks very alike the other panel as well. We look for the correlations of the bulk versus single cell variant allelic frequencies that we characterize, and we didn't separate between, but in this cancer, most of them are SMVs, and it shows a very good correlation. And again, because we look for try to stress the system, we went especially for the ones that we have very small percentage of tumor cells or of the cells mutated. So also we tried to do the first effort trying to reconstruct the tumor phylogeny in cases, especially we focus in the pre-malignant condition going to evolve into CLL. So this is the first combination. Again, in this particular case, we have 3 different ATM mutations and 1 P53. The first thing that we did is, and we have 2 different time points, all were observed in second time point by both techniques, but even by single cell, again, we were pleased to identify 2 that were below detection with our bulk pipeline or the pipeline for detection in bulk. But besides detecting, we were able to start building maps based on that. And the interesting thing is, in the same patient, looks like based on and we are using the algorithms from the company, but we're able to see that we have an ATM, a nonsense mutation where 3 independent subclones are generated, one with loss of heterozygosity in that particular one, but we have 2 additional subclones where they have the inactivation of the second copy of the gene. And more interestingly as well is we have a P53 mutation, but it's independent, it's not related to that particular clone.